So after learning a little bit more about the world, a lot of scientists had the question about what actually makes us us and how do our genes get passed on from our parents. So one view of this that prevailed was known as the blending hypothesis. And this is the idea that basically your genetic material from your mom and your dad blends together and then you are a mixture of them both. But what didn't really make sense was how our siblings could look different from us if this was true. So another hypothesis arose known as the particulate hypothesis, and this is the idea that parents pass on discrete heritable traits to us known as genes. And a monk who is also a scientist named Gregor Mendel documented this mechanism through his experiments with pea plants. So these pea plants produce beautiful flowers, and that is kind of what stemmed Mendel's original approach to identify the laws of inheritance. He discovered these basic principles of heredity by breeding garden peas in carefully planned experiments. There are several reasons why he chose to work with peas, but first of all, he chose to work with them because there are many different types of peas that have distinct features or characters such as their color, and these characters varied. So for example, you could have purple or white flowers, and he called these traits. And he also liked that he could control the mating between the plants, because what he would do is take trimmings of these pea plants, and then he would then, after taking the parents, take some of the pollen from the male plant and add it to the egg of the female plant, and end up being able to choose which plants fertilized which other plants. And then he observed the results. In his experiments, he chose to only track traits that occurred in two distinct forms. So for example, purple or white flowers, round seeds or wrinkled seeds, so that he could focus on the variables at hand. And he also chose to use varieties that were true breeding. So he wanted to make sure that he found offspring that were produced the same as the parents when they were pollinated with each other. In his experiment, Mendel would mate two true breeding varieties in a process called hybridization. So for example, he would mate a purple flower and a white flower and observe what happened. Those true breeding parents were known as the P generation. The offspring produced by the P generation were referred to as the F1 generation. And then finally, when the F1s were mated together, these produced a F2 generation. So he would take two of the offspring and mate them, and this would be the F2. What Mendel noticed is that when he crossed the true breeding white and purple plants, the parents, all of the F1, the first set of offspring, were purple. But when he mated offspring with offspring, he noticed that many of the plants still were purple, but some of them were white. And he realized there was a ratio of about 3 to 1 of purple to white flowers in this F2 generation. So he would have a purple parent and a white parent. He would cross them, and in the F1, all the plants would have purple flowers. And then he would mate one F1 with another F1, and he would notice that at a ratio of approximately 3 to 1, you would observe 3 purple flower plants to 1 white flower in the F2 generation. He seemed to observe that the heritable factor for white flowers was hidden or masked whenever there was a purple flower factor present. So he called this purple flower factor a dominant trait, and he called the white flower factor a recessive trait. And this factor for white colors was not diluted or destroyed, he determined, because it reappeared just as it did in the parents, finally again in the F2 generation. He observed 6 other pea plant characters, each of which could express one of 2 traits, and now this heritable factor we refer to as a gene. So these are the traits that Mendel looked at, and if you take a look at the F2 generation dominant to recessive phenotype, all of them expressed an approximately 3 to 1 phenotype of dominant to recessive. This model that he developed based on his experiments helped to explain why we observe the 3-to-1 ratio. And there are 4 concepts that make up his model. So the first is that alternative versions of genes are the reason behind the differences in inherited traits. So for example, the gene for flower color has 2 versions, either purple or white, and we call these alternative versions alleles. Each of these genes resides at a specific locus or location on a specific chromosome. So we know now, based on our study of DNA, that at the molecular level, a gene is a sequence of nucleotides that gets transcribed and then translated into a protein. And that protein has physical effects on our cells that produce the physical effects that we see. So, for example, the allele for purple flowers creates a sequence of DNA that codes for an enzyme that provides that purple coloring. Whereas the allele for white flowers codes for a different gene, has a different nucleotide sequence, and we don't get the enzyme when that gene is translated, so we get a white flower. The second concept Mendel discovered was that for each character, an organism inherits 2 alleles, one from each parent. Mendel did not know anything about chromosomes at the time, but he, based on his experiments, was able to determine that each of the plants contained 1 allele from each parent. The 2 alleles could be identical, such as with the true breeding parents, or they may differ, such as in the hybrids, and we'll take a look at what those alleles actually look like in a moment. The third concept was that if the 2 alleles at a locus differ, for example, if you have 1 dominant and 1 recessive, then the dominant allele determines the organism's appearance. So as long as there's a dominant allele present, you're going to observe the dominant phenotype, which is the physical manifestation. The other one, the recessive allele, will have no noticeable effect on the appearance. So with the flower color example, all of the F1 hybrids, the first generation of offspring, were purple because they all contained at least 1 dominant allele for purple flowers. Finally, one of Mendel's main laws is known as the law of segregation, and this is the 4th concept in his model. The law of segregation states that the 2 alleles for a particular character separate during gamete formation and end up in different gametes. This means that an egg or a sperm only gets 1 of the copies of the 2 alleles that are present in the organism. And this makes sense because we see this when the homologous chromosomes get distributed in meiosis to the different gametes when they are hacked. But remember, Mendel didn't know anything about chromosomes or meiosis, but he observed that the egg or the sperm could only contain 1 of the alleles, which makes sense because then when they're joined by another egg or sperm, you have both copies of the allele, 1 from each parent. This model explains why he observed the 3-to-1 ratio in the F2 generation of his crosses. But now we know, thanks to Mendel, how to model this using something known as a Punnett square. And with Punnett squares, we can predict the results of a genetic cross between individuals of a known genetic makeup. So as long as we know the parents, genotypes, we can determine their phenotypes. A capital letter is used to represent a dominant allele, and a lowercase letter represents a recessive allele. So for example, the capital P is the purple flower allele, and lowercase p is the white flower allele. Usually, the letter that they choose is representative of the first letter of the dominant trait. So for example, in the parent generation, we had true breeding parents, so purple with dominant, capital P, and white, which is recessive, lowercase p and lowercase p. We then see in the F1 generation, each parent donates 1 of its alleles, but because they're true breeding, it doesn't matter which one they donate. Purple is going to give capital P, and white is going to give lowercase p. So this creates a whole set of F1 flowers that have the genotype big P, little p. However, when these are crossed with each other, we are going to get a ratio where half of these purple flowers will donate a capital P to the offspring, and half of them will donate a lowercase p to the offspring. And so in a way, you can kind of do multiplication when you set this up. Big P gives us big P, we get a purple flower. Big P, little p gives us a hybrid, and we get a purple flower. Big P, little p gives us another hybrid, we get a purple flower. And then little p gives us a white flower, the recessive phenotype. Once again, you have 3 purples to 1 white. A few terms that might be useful when you're taking a look at these crosses. The term homozygote means that the organism contains 2 identical alleles for a particular gene, and that is said to be homozygous. So you could be homozygous dominant if you have 2 big P, for example, purple in this case, or homozygous recessive would be little p. If you have 2 different alleles for a gene, you're said to be heterozygous. So that would be, for example, big P, little p. Unlike homozygotes, heterozygotes are not true breeding. So you wouldn't use those as a true breeding parent. They're considered hybrids. Because of the effects of the dominant and recessive alleles, however, our organism's traits don't always reveal its genetic composition. So we have to distinguish between an organism's phenotype, what they look like physically, and their genotype, what their genes look like. For example, with the flowers, big P and big P, little p plants have the same phenotype. They're both purple, but they have different genotypes. So this shows us how our phenotype and genotype can differ. With our phenotypes, we have 3 purples here. You can see one of them is a big P, 2 of them are big P. This is the F1 cross. And then one of them is little p, which is homozygous recessive and is white. But notice our genotype ratio is 1 to 2 to 1, whereas our phenotype ratio, as Mendel observed, is 3 to 1. The next video will explain how you can determine the offspring's phenotype and genotype when you don't know the genotype of the parents, and that will lead us to Mendel's second law.